introduce our speaker today. So Zijun Gao is a tenure-track assistant professor in the Department of Data Science and Operations at USC Michelle Business School. So she received her PhD in the Department of Statistics from Stanford University in 2022, supervised by Professor Trevor Hasty. She served as a research associate in the Department of Pure Mathematics and Mathematical Statistics at the University of Cambridge from 2022 to 2023, hosted by Professor Zijun Gao. Her works focus on developing efficient methodologies in causal inference, multiple testing, and machine learning. She also works on real-world data-motivated topics with a specific emphasis on applications in business and healthcare. So welcome our speaker.
Thank you so much for that introduction. Do I have to speak into the speaker or is it
No, it should go through this. Yeah, so you should be okay right there.
So can basically dancing on the stage. Great. Thank you so much for coming. I know this is like right before the spring break. I actually scheduled my flight today, like around 8 p.m. So I will also be like pretty much looking forward to get this done and then start my like spring break journey, getting myself away from the teaching things. Okay. Don't tell my colleagues from Marshall. Yeah, I'm sick of teaching now. Anyway, so today, hopefully my talk will not be too boring and not too technical, so that it will not be the last straw on your back before you start to celebrate your spring break. And today I'm going to talk about something titled Selective Inference for Adaptive Trials using Conditional Randomization Tests. So this is work with my great host at Cambridge, Qingyuan, and also his previous PhD, but currently a postdoc at EPFL. So to talk about to start or kick off the talk today, this is my outline. So I will start by talking about the object, sort of like the adaptive clinical trials we want to analyze. And also I mentioned what are the challenges they're in. So why we need to study that, but not something that has already been investigated. And then we will propose our solution, which is the conditional randomization test. After that, I will talk about sort of extension or application of our most standard conditional randomization test to a case where we have a continuous biomarker. And after that, I will summarize the talk with some discussions and future directions. So if you have any questions at any point of the talk, please feel free to interrupt me and ask. I really love this interactive discussions like throughout the talk. All right. So without further ado, let me just jump in and tell you about what is adaptive trials. However, to talk about what is adaptive trials, I need to first review a little bit about the notations for a standardized or a standard clinical trial. So I draw this little diagram, maybe not spending the whole night, but took me a few hours to demonstrate what is the pipeline of running a clinical trial. So here, suppose we start with a pool of patients or units, and then we will randomly assign part of them to the treatment noted in the USC gold. And then the rest of them will be considered as in the control group, which is colored in the USC red or USC card. And after some time, we will observe or collect responses from all the participants and then evaluate whether this treatment is beneficial to them or not. And dating back to my PhD works, I was pretty much interested in investigating the heterogeneity in the treatment effect, meaning that some drugs may be benefiting to you and not working for me at all. And suppose that in this case, we do observe the treatment effect is heterogeneous. And this drug or treatment is actually only working for the senior group, but not the young guys. Anyway, young guys will gonna recover from, say, COVID. So in this case, our final conclusion will be, okay, this treatment is good for the senior group, and let's recommend that to older people once they go to see doctor for such a reason. Okay. So far so good. But one thing you may notice is, even though our final conclusion, our final drug recommendation only touch on the senior group, but there are quite a number of young people involved in the whole trial. So they're not benefiting in the trial, they're not going to benefit from once the drug is onto the market, but they're adding a lot to the clinical trial cost. So a natural question will be, can we do something better than this one so we can save our budget by not recruiting so many young people?
Sorry, I have
Yes.
a question, sorry, just because this is a test of heterogeneity, right? It's a test of the treatment by being old versus young, right? So it's different than the normal clinical trial being the test of the treatment only. So you're going in a priori with that as the main hypothesis, or that there's a subgroup that it's effective, or that you've done a secondary analysis for the effectiveness within the subgroup of it being effective in older people.
That's a good question. So the primary goal is to show that treatment is effective to the whole population.
Yes.
And then typically nowadays, people are not only satisfied with like benefit for everybody. They want to do like precision medication or want to personalize recommendation. So they actually will go into different subgroups and see whether this treatment is effective for them or not. So eventually the final conclusion or like a recommendation will be, okay, recommend the treatment to this, and this type of person rather than everybody. Right. So here I would say the primary goal is still to making sure that this treatment is effective. But beyond that, we also want to offer some conclusions regarding heterogeneity.
Of groups that you a priori know about?
That's a good question. So throughout the talk, most of the time, I will focus on the case that I have a candidate set of possible groups. For example, senior, young, or like female, male. But later on, I will talk about the biomarker case where the group is sort of like known beforehand, and then I need to learn the cutoff point where in a data-driven way. So the final subgroup will also be sort of adaptive or data-driven. Okay, great. Lovely. So I was saying that we have wasted some money on the young people because they're not going to benefit from this drug anyway. And then nowadays, like we got this like huge budget cut from our current government, which is a sad thing. So we need to be a bit more careful about how we spend money. All right. So how can we save a little bit money from how can we save money or making the clinical trial more economical? So one possible way or solution is running an adaptive clinical trial. So let me use the same diagram to show what it is being adapted now. So rather than directly assigning everybody to either treatment or control, we now only take a subpopulation out of it, like denoted here, and we call it stage 1. And we run a standard randomizing standard clinical trial within this subgroup, or say stage 1 patients. And then after some time, we'll be analyzing the responses from the stage 1 patients. And then at that point, we may already be able to find that only senior people are going to benefit from this treatment. Then for the next stage, rather than recruiting everybody else in the whole population, or say the pool, we will be only focusing on the selected subgroup, which is the old patients. So in the second stage, we will only recruit older people left in the pool. In the meanwhile, we also adapt the hypothesis that we care about. So rather than investigating whether this treatment in fact is beneficial to the population in general, we will be asking whether this is going to benefit the older people or not. All right, so by making this clinical trial adaptive, in the second stage, we do save some money by not recruiting young people in the stage 2. All right, so this is sort of like demonstration of what an adaptive trial could be. But this is not the only possible way of making it adaptive. So here, what are adaptive is? One, who we're going to recruit. And second, what are the hypotheses? And in real life, other adaptivity could come into the scene. For example, we can alter the probability of assigning the treatment to the people. Maybe previously were 50-50 and now we change it to 60-40. Or suppose there are different dosage of the same treatment, like same like vaccine. And then maybe we'll start from like 3 dosages and then we find one of them is ineffective. So we just drop it. That is also adaptive. So there are like multiple ways of making it adaptive. And all of those things will be and then we aim to develop a procedure that can treat those adaptive treatment clinical trials simultaneously or in a unified framework. All right. So to summarize a little bit about adaptive trial, so I want to use this little cartoon from a paper dating back around 20 years. So there, they plot the adaptive trial as a map. So the standard trial will starting from the oval and only having one path to the destination. And different from that, adaptive trials are having several paths from the top to the bottom. And which path you're going to take is guided by the complex called data. So basically, you use your use the data accumulated so far to guide which path you want to go for the next stage. And adaptive trials, maybe you have heard about it for some time, and then I will say it's not a new thing. It has been brought up maybe 50 years ago. But why we're starting to analyze it now? Because it has been in the discussion for 50 years, but it hasn't been really implemented or implemented massively up till now. And recently, or rather recently, FDA launched a manual or document for how to conduct adaptive trial. And there, after this, a lot of discussions have been occurred, and people are debating back and forth about whether we should be making adaptive trials or not. So there are a lot of like voices supporting that we should make trials adaptive. For example, I show by making it adaptive, we can save some money and we can only focus on the groups and potentially avoid some harms to the subgroups which are actually being hurt by the treatment. But there are also other concerns which making people reluctant to take on the adaptive trials. So for example, there are some logistic issues, like maybe we have to explain to the patients that we're making this thing adaptive. So our first recommendation will be different from our later recommendations, where patients may frown at and say, I'm not happy, like what are you doing in the backstage, blah, blah, blah. But as a statistician, I'm not super familiar with how to deal with the patients, but I'm familiar with some like statistical challenge here. And notably, one of the most prominent statistical challenge is a selection bias. So I will later talk about where the selection bias come into the being. But for now, I will just use one sentence summary to give you a rough impression of why there could be a bias due to selection. So recall that in the previous slide, I show at the end of the first stage, we do want to choose which subgroup we want to focus on. So this is where the selection happens. And if we just ignore the selection and use everybody in the trial to do the analysis, then there could be the double depletion. All right. So this selection bias thing will become more apparent as I talk about as my talk goes. All right. So next thing I want to bring into your attention is the randomization test, which is the second keyword in my title. And this is also the thing that we want to use to analyze the adaptive trials. So similar to before, where my goal was to talk about what is adaptive trial, but I started from a more humble attempt, which is a standard clinical trial. So this time I will again, in order to talk about what is computational randomization test, which is our solution, we start by a more humble or more old notion called randomization test. The randomization test has been there for, I think, almost 100 years, dating back to the very first, maybe the father of stats called Fisher, also from Cambridge. And then there are quite a few good advantages about randomization tests. And here I just list two, which could be more like appealing to my biostats friends. So the first one is the randomization test is modeling. It only requires about the assumptions on where, say, how the treatment is designed. Is it a randomized trial? Like, is it 50-50 or is it 60-40? And then we don't need any assumptions about, say, the modeling of the outcome, modeling of the covariates, and then like modeling about the dependence across units, whether they are IID or there's some dependence. And that's all fine. So the modeling assumption is becoming sort of important if we're dealing with a very complicated objects where we're actually pretty much unsure about the modeling assumptions. The second the second advantage of having this modeling randomization test will be we have the finite sample guarantee. So typically, when I was learning stats from my very first class, most of the properties I learned are based on CLT, which is asymptotic. And then when things are asymptotic, we always ask like when the asymptotics kick in. Is like a sample size 500 sufficient? Is a sample size 5,000 sufficient? Or if not, then what is the minimal number of data points or say units we have to get to deliver our like a statistical analysis? We don't have to worry about that. We have the finite sample guarantee. No matter you're having a sample size of 10 or 100 or like 1,000, our analysis or our statistical guarantee all apply. So as a implication, our analysis will be more favorable if we only have a small sample, which is usually the case when we're running some expensive clinical trials. All right. So those two are the advantages I want to highlight about, but there are also like other advantages. If you want to learn more about randomization tests, I'm more than happy to talk about it offline. Okay, and then as before I mentioned that randomization tests actually have a very long history, but it was sort of like not the out of the circle for quite like maybe 70, 80 years. And recently, it got quite popular, partially thanks to the conformal inference. Maybe you have already heard about this word. So I would say that conformal inference have a lot of similarity with a randomization test, but also some difference. If you're interested about the connections and differences between conformal inference and randomization test, feel free to read about this great review paper by my supervisor and also his previous PhD. All right. So now coming back to the adaptive trial analysis, I will show you first what is the common practice of applying the randomization test to the adaptive trial, or say part of the adaptive trial. So the easiest thing is let's just forget about what is happening in stage 1 and only apply the statistical analysis to the stage 2. So good thing is this is valid in the sense that the selection is solely based on stage 1. It has nothing to do with the stage 2. So we will not be susceptible to the selection bias. So how do we do that? We just throw away the stage 1 data and then go grab the stage 2 units. The randomization test will first reassign the treatment to the units in stage 2. So previously, the first one was treated, the second one was under control, and now we just reassign sort of like permute, and then the first one becomes the one that got the control, the second one becomes treated, etc., etc. And then we do this re-randomization, or say permutation, permuting the treatment assignment multiple times, and each time we can compute some test statistic depending on the reassigned treatment. And under the null hypothesis, saying that there's actually no treatment effect, all those test statistics are exchangeable. So we can use the test statistic computed on the alternative assignment to construct the null distribution, or say the reference distribution, and compare the observed test statistic T to the reference distribution to get the p-value. In particular, I find it's helpful for to talk about randomization or introduce randomization to someone who didn't come from such a background. You can just think about this is more or less a permutation test. And then in the middle stage, we're just like permuting the treatment assignment. And finally, we evaluate a bunch of test statistics for different permutations. And this permutation, the p-value is basically how the permutation p-value is constructed. All right. So this is the common practice where we are applying the randomization test, where you can think about it as like a permutation test to the second stage. So far, so good. We have the guarantee, and then we have some power. But the problem you may already become aware of is, I said at least 3 times that, okay, we only focus on stage 1 or 2, and then we forget everything about it. So that is actually not so data efficient. in the sense that we do have some senior patients involved in the first stage, and throwing away their data just makes them heartbroken. So can we recycle their data? Can we still like don't just put the stage 1 into the waste bin, or if it's already there, I want to take them back and see whether there's still some recyclable part information out there. All right, then maybe you say, okay, then just take everybody back. Let's just pull the stage 1 and the stage 2 all together and then run a randomization test or permutation procedure in your mind to everybody that is in this trial from the beginning to the end. All right, so procedure-wise, it's pretty easy to implement. We just like got everybody from the selected group, like older people, and then we just permute, and each permutation give us different a new test of statistical evaluation. We construct a reference distribution and compute a p-value. Procedure-wise, it's basically the same as what we did before, just like getting more people into the play or into the party. But this time we are subject ourselves to the selection bias. Because we know that in the first stage, we did some selection. And now if we just ignore that selection and do a vanilla randomization test, then we are actually using the first stage data twice. And that is what we call the double dependency. All right, so now we are being caught. We want to use more data, but if we use more data, then we're having the selection bias issue. So can we get something that leverages both stages and is free from selection bias? Well, that is our goal, and that is also the conditional randomization test I'm going to tell you about in a second that is going to achieve. All right, so now let me tell you what is the conditional randomization test. So in order to tell you how does that work, I want to motivate it to explain a little bit more about why the previous double dipping procedure does not work. All right, so this is basically the previous slides, but instead of just, but one more thing, one minor difference is that I pictured something in the middle, which is essentially the selection after the first stage. So recall that in the reality, we grab the stage 1 data, and then we make some analysis based on their responses, and we select the senior group to be the group of interest. And then we go ahead and recruit only people from the senior group, and we test that whether the senior people are benefiting or not. All right, so it's possible that if we reassign the treatment for the stage 1, and then we reevaluate whether reperform the analysis after seeing the stage 1 result, and we end up in a different selection, as depicted here. So in that case, we'll be selecting the young group as our group of interest. And then if that's the real case, or say in the parallel world, we will actually be recruiting a different stage 2, where we focus only on the young people. And more seriously, in that counterfactual world, say parallel world, we'll be testing a different hypothesis. That is, whether the treatment is benefiting the young people or not. So you can see that what happens in this world is not analogous or comparable to what happens in this parallel world. So when we construct the Snell distribution, we shouldn't include the things that are not comparable. In the sense that the test of statistics evaluated here is not suitable to be included in the histogram on the right-hand side. And because the previous case, the double dipping procedure, they include everything. And that is the reason why that is not valid. All right, so now our solution, or say our proposal, is pretty simple. I said that in this case, the test of statistics is not comparable. Then what should we do? We just describe that. So in the first stage, we just permute. And for each permuted assignment, we evaluate whether our selection will be the same as what we observe in this real world. If it is the same, like this, then we just continue. Permute the second stage and compute a t-star. However, if it's not the same, just as the counterexample I just introduced, that we select a different subgroup. then we just stop there and call it the end of the day. We will not proceed along this thread. So we do a bunch of these cases, collect a bunch of successfully accepted treatment assignments, and got a series of T-star to construct the null distribution. Finally, it's pretty much routine that we compare the observed tested statistic to the distribution constructed for only the accepted treatment assignment. And this p-value will be, I assure you, valid. All right, so this is pretty much our proposal, which we call it a conditional randomization test, because we can consider that as the randomization test condition on that we observe the same selection. All right, so this is pretty much the overview and also sort of this is pretty much the introduction of our methodology, the conditional randomization test. I will not go into more details, like I will not like shower you with terminologies like potential outcome model, like DAG, blah, blah, blah. But if you're interested in more technical details, I'm happy to chat offline. All right, so from here, I would love to introduce a few good properties of the procedure I just introduced, the conditional randomization test, which is short for CRT. So the first advantage is the validity, or say type 1 error control. In particular, we control some a stronger notion of type 1 error, which is called the conditional type 1 error. So that means we not only control the type 1 error, marginalize the overall possible selections. We say that among like specific type of selection, among like trials of selecting the young, among the trials of selecting the old, we all control the corresponding type 1 error control. So this conditional type 1 error control allow us to treat sort of like the selected group as pre-specified, because like this error control is restricted to that specific subset of the trials where possible. All right, and then a quick note is if we have the conditional notion of type 1 error control, we naturally have the marginal type 1 error control, which is a more frequently and commonly used metric. All right, so, sorry. I think the next thing I want to talk about is we have some power gain compared to the sample splitting procedure, where the procedure which only using part of this, which only using stage 2, basically not using data from all stages. And this is intuitively very straightforward because like throwing away data will usually hurt the data efficiency. All right. So, and then the third thing I want to talk about is this procedure is pretty flexible. It's accommodating all, or maybe not all, but most of the adaptive designs we're aware of. For example, I mentioned like we can drop a few arms, is fine. We can drop a few units, is also fine. And then we can maybe adjust the ratio of treatment and control group is also fine. Our work all adapt to those cases. And also we have offer flexibility regarding what test statistically you use. We can use some like difference in means, or you can use like your fancy like machine learning assisted W robust estimators. That's all fine. And also we accommodate almost any type of selection. For example, previously I said like selecting between young and old, we can also selecting between female and male, or as long as we have some like pre-specified possible selections, we can run our procedure. We can always run our procedure too. All right, so to summarize, we have 3 parts of the advantage. We have validity, we have some power, in particular compared to the sample splitting or stage 2 only procedure. And we have the flexibility regarding adaptivity, selection, and the test statistics. All right. So actually, this is the place where I actually can jump into the specific biomarker application. But I want to just talk a little bit about the computation challenge, just in case like some of you are interested in like designing some cool MCMC algorithms that I may, and then we may have something to talk about after offline. So just maybe a quick like 2 minutes discussion on the computation part. So previously I said like this randomization test is basically the permutation test where we just do a bunch of permutations and we're done. However, now I say let's do a conditional randomization test that about the permutation, we also need to verify whether the permuted assignment is giving the same or not the same selection. So that is sort of something built for rejection sampling. We just permute and we verify the selection. If it's the same, we accept. If not, we reject. So far, so good. It's super simple, like super easy to implement. Like maybe high schoolers can do this out of an hour with the help of ChatGPT, I think. And then, but the thing is, it could be that it's possible that the rejection probability will be very high. Like say, if that could happen if say our selection is very stringent. Like we just select someone to be of a specific age, a specific gender, specific race. In that sense, maybe only 10 possible assignment out of like 10 million is satisfying our selection constraints. So the rejection sampling will be basically rejecting all the time. So when that happens, we propose to use MCMC. And in particular, we design something called random walk metropolis testing, which allow us to explore the accessible treatment assignment more efficiently. So I will not talk about like the specific details about this, but I want to give you the idea of why that could be more efficient. So it could be more efficient because the rejection sampling, every time it's starting from a fresh new treatment assignment. It has nothing to do with the previous accepted treatment assignment. However, for the MCMC, we will be starting from some acceptable treatment assignment and then move a little bit away from it, not too much. So we're still like pretty much similar with the previous acceptable one. And usually the constraint being if the previous case is satisfying the constraint, then its neighbor is quite likely to satisfy the constraint. So that's why by making a little bit of deviation from the previous acceptable candidate, we will with a higher probability accepting the next proposal. So we're going to see a way smaller chance of rejecting the hypothesis, rejecting the treatment assignment if we are running this. And we have some simulations showing that this is actually in the high rejection rate case, the MCMC is actually saving our life. All right, so that's pretty much about computation part. I just want to mention the high rejection thing and also some designing up and the design of MCMC algorithms. So finally, let me just demonstrate or try to convince you that our procedure is actually having some real power in the sense that it's actually working on some real data. So I crafted a hypothetical adaptive trial from a standardized clinical trial, meaning that the original data is actually from a standardized non-adaptive trial, but I create like stage 1, stage 2 by using its original data. So I only like sample it. I didn't really check their outcomes or covariates at all. All right. So the design is pretty much similar to what I showed in the diagram. So we have a randomized trial, and then we have the outcome to be a binary outcome, a binary indicator, and then the possible selections are either the participants are belonging to a senior or young group. So in the first stage, I subsampled the 400 units without replacement from the original data set. And then I performed some selections regarding which age group I should focus on. And then for the second stage, I will just zoom in the selected subgroup and then recruit another 200 units, which are not in the first stage. And that's pretty much it. So for the inference methods, I was comparing three types of randomization based methods. The first one is is basically the double dipping one. The second one is the data throwing one. And the third one is our procedure. And of course, we want to show that our procedure works better than the previous two. So the first thing is about validity. So here I show the p-value curves for comparing the three randomization test methods applied to this hypothetical adaptive trial. And then if the p-value is valid, then it should stay below this 45 degrees line. And you can see that the red curve representing to the double dipping method is crossing the 45 degree line. So it is actually invalid. There is the type 1 error inflation. And then the next thing is about power. So since the double dipping thing is not controlling type 1 error, so I kick it out in this comparison. So I will be only comparing our procedure to the data throwing strategy. And you can see that and now this time we're looking at the p-value and then we a fast increasing CDF will be indicating a more powerful procedure. And our procedure indicated in cyan color is shooting at a faster rate than the data throwing strategy, saying that we have at the same with a higher probability, we're going to reject the null in this case, enter the alternative. All right. And then we observe that if the selection is not so stringent, then our improvement compared to the data throwing strategy is more substantial. All right, so that's pretty much about the general conditional randomization test. So now I'm going to spend about like maybe less than 10 minutes to talk about a specific application of our CRT to the case where we're dealing with a continuous biomarker. In particular, there are two specific cases of characters about this application. So one is this biomarker is continuous. So if we just determine the group of interest to be the biomarker larger than a value, then there could be infinite possibility of that cutoff value. And second is previously I usually come up with like a pre-specified selected subgroups. But here is we only have a biomarker. We don't really have a specified subgroup of subgroup candidates. All right, so let's see how our procedure can be extended to address those two new challenges. So I will start by talking about how the biomarker kicks into this adaptive trials, and then I will talk about how to modify our procedures to address the issue. So this is like a diagram to show that how usually biomarkers are used in the clinical trial. So when we just do the clinical trials and then also collect the information about the biomarker. Then we do some data analysis and then we come up with how the treatment effects depend on the biomarker. From there, we will define like a specific cutoff. And we will think the people with the biomarker larger than the cutoff is going to benefit, and whose biomarker is smaller than the cutoff is not going to benefit or even hurt by the treatment. So in the future, we will only be assigning this type of drug to the ones that are having at least that number of biomarkers. All right. So if we're running adaptive trials, then this is pretty much with the biomarkers. Then this could be one possibility of making of using the biomarker information. In particular, in the first stage, we will be doing the selection. So we will be select some cutoff for the biomarker. So in the second stage, we adapt our hypothesis to be only care about people with the biomarkers larger than that cutoff. And for the people we recruit, we only focus on the subgroup with at least that number of biomarkers. Right. So generally speaking, the picture is the same as before. We just replace the old and young by the biomarkers value larger or smaller than the cutoff. But this cutoff is learned from the data. It's not pre-specified. All right. So now let's talk about how to do randomization inference for this type of case with the biomarkers. All right. So as before, we can always do the case, always do the data throwing case that we forget about stage 1 and we focus on stage 2 patients. So that's already done. Like everybody knows like how to do it. So I skip it. And the next thing is, what about our procedure? Okay, then you may ask, this apply your procedure. You said your procedure is pretty flexible. Then why can't you apply it to this case? Well, I can. The problem is it will barely have any power. So what's that? Let me explain. So so recall that we want to only take into the consideration of the treatment assignment, which is giving the same selection. And in this case, it's giving the same data-driven biomarker cutoff. And notice that the cutoff is actually a function of the first stage treatment assignment. So it's possible that different treatment assignment will give a different biomarker cutoff. So in that case, if we reassign the treatment, then all of them will come up with a different recommended cutoff. And all of them have to be discarded because we want to condition on exact selection event. So by the end of the day, yes.
Is the biomarker sort of a baseline biomarker before treatment, or is the biomarker responsive to treatment?
Baseline. Yeah. And then in this case, we will be only observing one data point in our histogram. So, and then the p-value will always be 1, and there will be absolutely no power. Okay, so the problem here is this selection is too stringent. It's almost impossible to yield the same biomarker cutoff by same biomarker cutoff by reassigning the treatment from the starting state.
Sorry, another
Yeah.
question here. Are we assuming that the biomarker has a threshold effect or that it's a continuous effect and you're just choosing, given the data, a threshold that you're assigning to this design? Does that make sense? Like in reality, the causal mechanism is that there's a threshold. And we know what that threshold is to that biomarker, right? So if you go back here, right, you have a cutoff. Right?
Yeah.
So I guess the question is, why have a cutoff and not just have a continuous biomarker that shows the probability of response is dependent on the biomarker level?
Oh, that's a good question. Maybe
And then have like your sampling be dependent on that probability.
I see. So, when I was thinking about this, biomarker application, actually, our procedure where the cutoff makes sense to me, at least, as long as the treatment effect is increasing regarding to the biomarker value. It could be like increasing like continuously or smoothly, or there could be some jump.
Okay.
And then like this case, absolutely, we want to stop here, like assigning treatments to those units. But for this case, I think people still want to have like cutoff.
Okay.
Because this related to like some like discontinuity design, blah, blah, blah. And they think like, okay, we will just assign treatment to the benefit, to the units who are going to benefit at least by that much.
Okay.
All right. Great. Okay, so now coming back to the case that our procedure simply applying it to the biomarker to the case with a continuous biomarker will not have any power. It will be valid, but p-value is always equal to 1 who want to use it. So next is how are we going to address the problem of no power? So our solution is actually doing something regarding in the selection procedure or selection step. So previously I was saying that, okay, you can like learn an arbitrary treatment effect function depending on the biomarker, and using that learned treatment effect function to determine where is the cutoff. For example, comparing that treatment function to some specific pre-specified value like 0 or 1. But in this case, we're saying that, okay, you cannot have so much flexibility in choosing your selection criteria. So, or like your learned treatment effect function as the biomarker, as a function of the biomarker. So let's do the selection part in this way. So we will arrange all the units according to their biomarker value from small to large. So we're expecting that the treatment effect is also increasing from low to high. And then we will sequentially review their treatment assignments. We will first review the assignment of the first one, and then second and the third. And then basically in the increasing order of their biomarker as well as their treatment effect value. All right. And then at some point, we review, like, say, a bunch of their treatment assignment, and then we evaluate or estimate the treatment effect at the largest biomarker reviewed so far. For example, if we know the first 3 units, we aim to estimate the treatment effect at the third unit, which is associated with the highest biomarker reviewed so far. All right, and then we will continue this reviewing procedure until at some point we show that, okay, the treatment effect estimated at the largest biomarker review so far is positive. Then we stop there. And then the rest of the units, which we haven't touched so far, will be the subgroup of interest. Or say the largest biomarker review so far is our cutoff value. All right. So below this diagram, I'm trying to show like what happens in this like iterative procedure. We review 2, we estimate the treatment effect value here is still negative. Then we proceed, review another 2, and then now the treatment effect at the 4th person is positive. Then we stop. And then the first 4 are considered not of interest, and the last 3 are the subgroup that we want to investigate. So that this type of selection we want to see. And then we still have some flexibility, like we can estimate the treatment effect here in an arbitrary way you want, as long as this is done in such a sequential manner. All right, so the next thing I want to claim is if you agree with me and adopt the selection in the way as demonstrated in the previous slide, then our procedure will make no discussion at all. That means it will always yield the same cutoff. So this is basically just the treatment assignment without conditioning, or in the sense that we condition on something that is always satisfied. All right, because there's no discussion, then we will have the power, and then we can just do standardized randomization, conditional randomization test, or say randomization test, because conditioning is not effective here. Or in your mind, it could be just like permutation test. All right. So that's pretty much about the procedure regarding the case where we have a continuous biomarker, and then we're learning a data-driven cutoff to determine the subgroup of interest. So here I just quickly show you an example where we have a trial which involves a biomarker. So this is a GBCS trial where they're studying whether a hormone treatment is effective to the breast cancer or not. And the biomarker here is a type of receptor, and then they believe that if a patient have more receptors, then they can absorb the hormone better, so that the treatment effect is larger. So we applied our procedure in contrast to the case where they throw away a bunch of data to the GBCS trial. And we will find that both of our method and their method, and then the data throwing method selected patients with at least 5 receptors. And the p-value in our case is 0.003, and then in the case where they throw away some of the data, is only 0.024. So we do have a bit more power here. All right, so that's pretty much about the application to the biomarker case. So let me quickly wrap up. So what I did talk about today is we want to analyze the adaptive trial where the adaptivity brings in the selection. So we want to achieve a method which does a model inference without throwing the data from any stage, and also avoid the selection bias. So we introduced it to achieve that using conditional randomization tests, and then we illustrate some advantage regarding validity, power, and flexibility. So for the future works, the first thing we're currently considering is using the data-driven biomarker. So this is different from a data-driven cutoff. That is, in that case, we already know the biomarker, and we want to determine where we want to stop. But for data-driven biomarkers, like we actually have no idea which covariate or which biometrics should serve as the biomarker. And maybe we want to choose one out of them, or we create, say, a combination of some biometrics to serve as our like pseudo or artificial biomarker. So that's what I mean by data-driven biomarker. So that will be like, that's actually used in practice. For example, like some gene scores are basically the gene information, the fusion of a bunch of gene information. Right, so that's the first part. And the second part is about power. So we do see a lot of power gains compared to the case where they're throwing away the data. But we didn't really have a very concrete idea about what is the most powerful test statistic to choose for our conditional randomization test. Actually, what is the most powerful statistic for a randomization test is also not well studied, and currently that is one of my like focus or say in research interest. Happy to talk more about that offline. And then the third thing is about computation. I know that biostatisticians are actually much better regarding the computational part of statistical methods. So that's why I want to highlight this in my talk here. So if you are experts on MCMC or you're interested in developing like the fast conditional randomization test, I'm more than happy to talk about it. All right. So more broadly, personally speaking, I think this adaptive clinical trials is actually pretty promising. It has a lot of like benefits or a lot to offer. And then there are, say, not so many adaptive trials so far that I can access. So if you have some like adaptive trial data that you would love to try our methods on, I will be like super excited to jump in. My advisor at Cambridge, he's now working with a KPMG, a consulting firm, to do their first adaptive trial. So basically, they want to understand how COVID affects like how people work, and then they want to do it in a randomized trial and adaptive manner. And they're just starting to write the protocol. So hopefully, they can get it done by 2 years, but that's quite a long time for me. All right. So adaptive clinical trials is promising, and we need more sub data and hopefully and more analysis associated. The second thing I want to mention is heterogeneity or heterogeneous treatment effect in clinical trials. So heterogeneity is the key word of my research interest in the causal field. So I have heterogeneity for estimation, for example, policymaking, etc. If you're interested, I'm also happy to talk about it. And then this time I focus on the heterogeneity in clinical trials, and that's the motivation why we're having this adaptivity, because if there's no heterogeneity, we don't need to adapt. So finally, it's sort of advertisement for modeling randomization inference. This is taking on at least in conformal area, like black box based model inference follow-up, but I think it also has a lot to do, or a lot of potential in the clinical trial. All right. So with that, I have concluded my today's presentation. Thank you so much for coming, and thank you so much for staying to the end. Hopefully, I'm not too much over the timeline. Okay, I still have 5 minutes for questions. All right, so that's pretty much it. If I have any questions, I think this could be a good time to ask.
Any questions?
Yeah, so for your future work, the first one is biomarkers not pre-specified but learned from data.
Yes.
That can be seen as a inference condition on the clustering of some variables in the data.
Yes, exactly.
Then on that cluster, we put some inference about the outcome.
Yes, yes.
Okay. Yeah, I think there are a lot of nice things for that.
Yeah, I think for a specific case you're talking about, it's also quite hard for us to do a direct conditioning, because usually if you perturb the data a little bit, then the cluster will not be exactly the same.
Yeah.
Well, in our case, we want to condition on the exact selection. So basically that clustering.
All right.
Right. So I guess that may come to something similar to the biomarker case, where we say naively apply our procedure will give you no power. But maybe some clever way of doing clustering, so that in our case is we will discard nothing if we're doing that type of selection, then that will give you some power as well as the validity.
Yeah,
Right.
yeah.
That's a very interesting case in the sense that I also have a collaboration with the Cambridge biostats. They call MRC unit. This is basically their biostats branch. And then they were doing a lot of clusterings regarding brain cells. And then they have this and that request about clustering. So I got to learn a little bit about it. And I think that there's a lot unknowns about the clustering. So if you're experts on that, I'm happy to talk more about my other researchers further to relate to clustering and also this one. Yeah, hi.
Can I ask a big picture question? So I'm not a clinical trialist, but I'm contemplating this idea of adaptive clinical trials.
Oh yeah,
And
welcome.
it seems like, in stage 1, typically I would assume that the clinical trial is powered to detect that main effective treatment. And it seems like this is kind of an interaction question they're asking at stage 1. Are there problems with being underpowered to detect that interaction and using that interaction to determine stage 2?
Oh, that's a good question. Yeah, I totally agree that for example, stage 1 is usually smaller or exploratory. So maybe there's not sufficient or not enough evidence saying that we are able to focus on one subgroup.
Yeah.
But if that's the case, it's fine. Then we just recruit everybody into the second stage. I think that adaptivity will kick in or be helpful if there is really some huge heterogeneity.
So it's got
Yeah,
big
in the
interaction
effect.
effects.
Yeah, or maybe there are some harms. For example, young people are being hurt by the vaccine. So there are some rumors saying that COVID vaccine can hurt young people, something like that. If that's the case, then definitely being adaptive can avoid harms.
And similarly, I guess, if there's some people in the trial who maybe are slow responders to the treatment or something, seen later on, this really is effective for subgroups that have a strong effect early of the treatment rather than slow responders.
Yeah. I feel nowadays people are thinking about doing this hierarchically. For example, some are responding really fast and really strongly, then maybe they can first go into a stage 2. I would say a larger proportion of them will come into the stage 2. And then for the low responders, maybe they will be kept under observation for a longer time. So people will say, okay, stay in stage 1 for a bit longer, and then until we're confident that you're benefiting, then we will release you to stage 2. So they do that hierarchically at different time points. Yeah.
Thank you.
Any other questions? Just to let everybody know, we are meeting afterwards with the speaker in the Biostats Lounge. If people would like to join us there, that would be great. We can continue this discussion. All right.
Thank you so much. on a rainy day and right before the spring break.